Okay, guys, this is the beginning of exam 3. I've already begun your exam 3 as far as writing it up. So we're going to start off with you guys reading, I believe it was chapter 12, genetic engineering. So here you have the slides you should have them at home. The objective here is to show you how a gene, and by now the definition of a gene should be self-explanatory to you, all right? A segment of the DNA that has a specific code for a certain enzyme that is going to be expressed, all right? But the reality is, and many of you may not know this, but this has been going on for 25 years, all right, where we can actually take genes out of humans. For example, let's say the human insulin gene, all right? And then what we could do is we could take that human insulin gene and put it into a bacterium and allow the bacterium to express the protein. And in doing so, we can then take that protein, purify it, and put it in a bottle and sell it to people that need it to inject themselves with insulin. So we don't have to take insulin from humans. We can actually have bacteria make human insulin for us. And I'm going to show you that process in a few minutes. All right. So the objective here is to show you how a gene might be cut out of a human DNA organism and inserted into what is called a plasmid. So before I use terms like plasmid, you have to understand what a plasmid is, right? So let's take a look at a bacterium or a bacteria, and here's a bacteria here, okay? And you can see that this bacteria has two sources of DNA, and one of them are its regular bacterial chromosome, which contains numerous amounts of genes, and the other one is what is called a plasmid, which is also circular, right, and also contains genes. So it has two sources of genes. All right. Here's an actual microphotograph. This is 1 micron, which is very, very, very small. You cannot see that with a regular microscope. All right. So what I want to focus in on are these plasmids that we see here. All right. These plasmids are small circular pieces of DNA that can occur in bacteria. Right. They replicate easily. That is to say, the bacteria replicate easily. That is that about every 20 minutes, a bacteria will replicate, and that's why if you leave food out for a long period of time, you'll find out it decays pretty quickly. And the reason is because the bacteria are exponentially replicating itself every 20 minutes. So before you know it, you have millions of bacteria on a piece of food that you have left out, whereas the cold, when you put it in the refrigerator, slows down that replication. All right? So they replicate easily, and the bacteria and any genes they contain are expressed, and you should understand that term expressed by now. Expressed is when you have a gene, and when you want to actually allow that gene to make the product, that is to say the protein or the enzyme, all right, it expresses that gene into a protein or an enzyme, all right? And these plasmids have genes just like those that are on the bacterial chromosome, all right? So let me move on to the next slide. Oh, I have to go there. So today what we're going to do is we're going to learn some terms that are going to be on the beginning of your exam 3, which will be coming up pretty soon. And also some of these questions will be replicated on your final exam. All right. So plasmids, and you can actually do this nowadays on a kitchen table. All right. Plasmids can be removed from the bacteria and then modified to suit human purposes. All right. And again, we've been doing this for a long time, and then inserted into a fresh bacterial cell. All right. And once they've been inserted back into that fresh bacterial cell, all right, they function as though they've been there all along. They don't know the difference. That is to say, once we take this plasmid out, all right, modify it, put it back into the bacteria, all right, it'll go along its regular lifespan, expressing the genes that are on its bacterial chromosome and expressing the genes that are in the plasmid, which is a circular piece of DNA. All right. So the bacteria will replicate itself along with its genes and the newly inserted human gene. Because what I'm going to do is I'm going to show you how we could actually take a human gene, like I said, for example, a human growth hormone, or for example, an insulin gene, or any other gene, a lactoferrin gene, something like that, and actually take this plasmid out, put a human gene inside of it, put that plasmid back into the bacteria, and then allow that bacteria to express all of its own genes, which means it'll make the proteins of its own genes, and also it will express the the genes that are in the plasmid, including the human gene. It knows no difference between its own genes and the human gene that we have inserted into it. And I've done this many times over the years, and it always works out very well. All right. So the bacteria will replicate itself along with its genes and the newly inserted human gene, which I just explained. And when the protein of the human gene is expressed, all right, along with the bacterial genes, it can be purified and utilized as a human protein, all right? And that's been done for, I don't know, 20, 30 years already. This is nothing new. Matter of fact, if you need, like, for example, human growth hormone, which is sometimes given to children, or many other different proteins, insulin, for example, is one of the big ones where a lot of people are diabetic and they need to inject insulin. Well, where do you think we get this insulin from? It's actually a protein that is often, if not all the time, we get from plasmids, right, and bacteria, and they can express a human gene, and that human gene or human bacteria is exactly the same as ours. There's no difference. All right. So let me move on to the next slide, because I want to try and cover as much as I can. All right. So let's take a look at this. We already went over some of this in the previous slides, right? So I'm going to start introducing some new terms to you, all right? And I can guarantee you that these terms will appear. I have writing all over my face. These terms will appear on your exam. So these are definitions that you're going to have to recognize and understand and be able to connect the dots, so to speak, all right? So one of the useful characteristics of a restriction endonuclease, and a restriction endonuclease is the same as a restriction enzyme. It has two names, all right? So if you hear restriction enzyme or you hear restriction nuclease, endonuclease, they are the same thing, all right? It's an enzyme, and as you know, enzymes are proteins, all right? And a restriction enzyme basically has the capability, right, to cut DNA at a certain sequence. So what do I mean by that? I mean, let's say, for example, you had a piece of human genome or any genome, all right, and you had sequenced it, which we've been doing again for about 20 years, and you found a certain gene in there that you wanted to cut out, all right? And so you could actually get what is called a restriction enzyme. You can look them up online. And you can pick out what enzyme you would like to use to cut out at a certain point in the genome, all right? That is to say, you want to cut out at a certain nucleotide sequence, right, that is not within the gene itself. Otherwise, you'd be cutting in the middle of the gene, and you don't want to do that. You want to cut the gene out. So here's one example that's given in your textbook, which you should be reading, by the way. You should be reading the textbook. You should be looking at my lectures online. You should be taking the sample exams that I'm putting online for you, and then you'll be prepared for the exam. All right? So here we can see, for example, a piece of DNA. All right? Now, they're just showing you a little spot in the DNA, right, where we might want to cut. All right? And there's a guanine, a cytosine, an adenine, a thymine, an adenine, a thymine, and thymine, and adenine, and so on and so forth. Right? So let's say we have this piece of DNA, we want to cut it. So that is our restriction site, right? That's where we actually want to, our restriction recognition site. This is where we want to cut, let's say, all right? So what we do is we take a restriction enzyme, which I just described, okay? And we can cut the DNA into two fragments, all right? So here's that piece of DNA. Now realize that it's double-stranded, as you can see, and it goes all the way down on both sides, but we want to cut it somewhere in here. So it's very easy. You can go online, and you can say, well, I want to find a restriction enzyme that cuts between the G and the A in this particular sequence, right? And that would be also between this G and A on this side of the strand. So we want something that might want to cut something like that. And most of them do cut like that. They cut like, you know, down between a G and an A, and then down between a G and A, but it has to be that particular sequence, all right, when you order that restriction enzyme. All right. So the restriction enzyme cuts the DNA into fragments, and it's really easy to do. You could, like I said, you could actually do this at home nowadays. You buy a little kit, you buy the restriction enzyme, you get a piece of DNA that you'd like to cut, all right, and you could do it. It's very easy, all right, if you know what you're doing, all right. So here you go. We go and we cut it, and sure enough, we cut this DNA, and we wind up, as you can see, it cut here between G and A, and here between the A and the G, here, here, and here. All right, and here it is, here, here, and here. All right, so now we have two ends of that piece of DNA. All right, and what's important is the terminology here. All right, the terminology is we have a name, and this is kind of a strange name, but it's actually a scientific name, and this is called what is called a sticky end. Sticky end, and the reason it's called a sticky end is simply because this adenine wants to base pair with a thymine. See, it is where the thymine was before. All right, this adenine wants to base pair with a thymine. All right, and this thymine wants to base pair with an adenine, and this one wants to base pair with an adenine as well. So they're kind of sticky in the sense that they're looking to get back together. All right, so this piece and this piece is kind of looking to get back together. So if we actually took another gene and cut it out with the same restriction enzyme from another source, all right, that's the important part, another source. Like, for example, over here, here is a piece of DNA that's been cut out, and you'll notice that it has sticky ends on both sides, all right? So you could also notice, maybe from this picture that's in your textbook as well, that we can actually take this outside gene, maybe this is the human growth hormone gene, maybe this is the insulin gene, and we can actually take this and insert it into the genome of the organism that we just broke open, all right? And you can see it has the correct sticky ends. For example, AAT, all right, T. And now you can see that A would stick over here, that A would stick over here, and those T's would be over here. And on the other side, the same thing would occur, right? So we have now the addition of the DNA fragment from another source, and here is that other source right here, okay? And now what we can do is we can use what is called an enzyme called DNA ligase, all right? DNA ligase is another enzyme that you're going to probably want to know the name of. And what it does is it makes those bonds, all right? The bonds that we talked about when we had in school, all right? These covalent bonds, it allows these covalent bonds to be made between the molecules in this direction so that we can actually get this new gene. As you can see, this gray one here is the new gene, and insert it into the old genome, all right? So let me read what I have here. One of the useful characteristics of restriction endonucleases or restriction enzymes is that they cut DNA in such a way that one of the two strands is left longer than the other, all right? You can see what I'm saying? Here's the two strands, and one is left longer than the other, and here's the other strand, one is left longer than the other, all right? This creates what is called a sticky end, all right? Because the longer strand could form base pairing, like you see here, these bases base pairing with these bases, because the longest strand can form base pairing with the single strand of the other DNA being this one, all right? Molecule that has the same kind of cut, and that's important too, all right? Now, one thing you don't want to do, and you're going to see maybe in the next slide, is you want to make sure you don't cut inside the gene that you want to take out of the other organism, all right? Otherwise, you will destroy that, and I'll show you why in a few minutes, all right? So this is in your textbook. You should read it. There'll be probably a few questions on your exam about it. I'm going to just move the slides. I have another computer over there, which is actually sending the image to my overhead projector to the screen, and then this computer right here is what's recording everything. All right. All right. So there are many different restriction endonucleases or restriction enzymes. All right. Each will make a cut at a particular sequence. In other words, each restriction enzyme has its own restriction site. So when you find a piece of DNA you want to cut, let's say here is a piece of D, this yardstick is a piece of DNA, and it has a gene in it, and you want to cut the gene out. The gene is between here and here. You want to make sure that the sequence that's inside the gene is not where you're going to cut. You want to cut outside of the gene, all right? So you'll have your sticky ends on both ends of that gene so you can put them into another genome. All right, so these enzymes have names. All of these restriction enzymes have names, and you may want to know where these enzymes come from, and that may also be a question on your exam. All right, and these restriction enzymes come from multiple organisms, mostly bacteria. All right, because bacteria have their own genome, they have their own DNA. All right, so they have to be able to protect themselves. So what they do is they make these restriction enzymes. And they make restriction enzymes that will cut DNA that is not in their own genome, right? And the reason they do that is because if it's an invading piece of material or an invading organism, right, it will have a different DNA sequence than theirs, and they make restriction enzymes that will cut foreign DNA. All right, so these enzymes have names, and they are derived from the scientific name of the organism that produced them, right? And they have very strange names like BAMH2, BAMH1, Hindi3. There are a whole bunch of numerous different types of restriction enzymes that can be purchased that are produced by bacteria so that they can destroy the DNA of other foreign organisms, right? So we already have seen this, so I'm going to move on to the next slide. All right, here's a pretty important one. I know this is in your textbook, and I can almost guarantee it's going to appear on your exam, exam 3, that is, right? So here I'm going to show you, or I'm going to take this a step further, so to speak. Here we have, let's say, for example, a human cell, all right? And you can actually do this with a cotton swab. Maybe you've seen this on TV, some forensic file or something, where they actually take a cotton swab and they swab the inside of the cheek of a certain individual. And each cell has its own genome inside of it. So in the nucleus is a chromatin, and in the chromatin is all the genes. So when you do that with a cotton swab, you can actually take a few cells and you could break down and take the DNA or isolate the DNA out of that particular human cell, right? Now, I did gene sequencing back in 2000 when this was brand new, and when I did my research, I had my own lab that was funded by the National Science Foundation and a few other places, and I did research in forensic science for about 3 years while I was writing my thesis and while I was teaching and so on and so forth, right? So this was pretty new back then, all right? So what you could do is you can isolate the DNA from the human cell, right? And once you sequence the genome, you can find the genes. So let's say I want to find this gene of interest, which I'm going to call the human insulin gene, all right? So here it is here, right? So it's very simple. Once I sequence this all out, I make sure I cut this human gene out so that I don't cut in the middle of that insulin gene. I don't want to cut in the middle of that insulin gene because you'll destroy it. So I'm going to cut to both sides of it. I'll find some sequence that does not appear inside the gene and cut it out. And here is my gene of interest now cut out, right? And here you can see, for example, there are those sticky ends that I spoke about just a few minutes ago, all right? All right, so now I have the human insulin gene right there with its sticky ends. Now I go back to my bacteria, really easy to do, and I remove the plasmid. And you say, well, how do you do that? There's just a few chemicals that you use. You use a little test tube, 1, 2, 3. You can do it on a kitchen table nowadays, like I said, and you can remove that plasmid, all right? So what you do is you cut that plasmid, right, with a restriction enzyme, all right? The same restriction enzyme that you would use to cut out the human gene, which will give you the same sticky ends, but in the reverse of the one you cut out. So here's the sticky end, right? And you can see this circular piece of DNA, right? The plasmid DNA, right? Now treated with that restriction enzyme has now been broken open, all right? Been broken open and has those sticky ends. Now it's as simple as this. You take the plasmid, you take the gene of interest. In this case, I said it was, for example, human insulin gene, right? And you put them in a test tube together with a little bit of DNA ligase, right? And you ligate the gene of interest, which is a human gene, into the bacterial chromosome, the plasmid chromosome, and there you can see the sticky ends will fit, right? And then, as I just said, you add DNA ligase, which is, is an enzyme, because it ends with ASE, which closes the circle with covalent bonds, which I mentioned just a little bit earlier. So now we have what is called a recombinant DNA plasmid. Recombinant DNA, which really means what we did is we made a new combination of DNA in that plasmid, all right? Recombinant DNA plasmid, and this recombinant DNA plasmid, all right, now has the gene of interest in it. That means that when it's put back into a bacterium, which is what you're seeing here, all right, when the time comes, which is pretty much every 20 minutes, all right, when the time comes, this bacteria will not only replicate itself, it'll replicate both its plasmid and its circular DNA. And when it goes to express the genes on either one of these sources of DNA, it will also express the human protein, all right? And if that human protein happens to be insulin, it will, again, express insulin. So all of these different proteins will come out sort of like oozing out of this bacteria, and one of them will be insulin, and that'll be sent to a lab where it'll be purified, put into bottles, and sent to a pharmacy, all right? And here you can see, for example, these cells now when they clone, and I really don't like that term clone because a lot of people have associated it with something completely different. But cloning simply means you're making a copy of the original, right? Now this is the original, so when it reproduces itself, right, every 20 minutes, it makes a copy of itself. Or you could say it's cloned in a sense, right? Right? And then what we can do is these genes or proteins are then isolated, that are expressed, right? They are harvested, and they can be used for a lot of different things. They can be used for anything that a human protein would be used for, right? Now, we just can't go around slaughtering humans to take some insulin from them so that we can save other people. So we use bacteria to make human insulin. There's no difference between it, all right? And there's a lot of different uses, all right? We modify our food products that way. We have certain heart medications, for example, and so on and so forth. So let me take this to the next slide, and make sure you do study this because I'm telling you now, all right, this is going to be on your exam 3. I've already started to write it. As a matter of fact, I'm starting to put notes up on the Canvas right now. I shouldn't say notes, but they are little mini tests that you can be taking, all right? So that you can see how well you're going to do on exam 3. And don't forget your final exam is your cumulative exam, and everything from exam 1, 2, and 3 will be on your cumulative exam when that time comes. All right, so let me go over a couple of things here, because I'm going to stop in a minute. All right, I want to go over some of the terms that we just used. All right, recombinant DNA simply means combining nucleotide sequences from 2 different sources. All right. Plasmids, again, now you should be familiar with plasmids, small circular DNA molecules that replicate separately from a much larger bacterial chromosome, all right? Restriction enzymes, or a restriction endonuclease, all right? Enzymes that protect bacterial cells against evading DNA from other organisms. I explained that, all right? And bacteria are a major source of restriction enzymes, and that you should understand by now as well. They make a lot of different types of restriction enzymes that cut various types of DNA, and all of that DNA is DNA from invading organisms, right? And the restriction site, well, that's the DNA sequence that is recognized by a particular restriction enzyme. All right? Let's see, we're into about 24 minutes. I'll go maybe one more slide, and then I'll post this one for you for tonight, and you should read these as often as you can. Or look at these videos along with the notes. And I have your notes published already. You should have a copy of them. They are on the system. So, well, I guess I'm not finished with the definitions. All right, restriction fragment. All right, ends of DNA pieces that are left after cutting with a restriction enzyme, also known as sticky ends. All right. DNA ligase, all right? So I'm kind of repeating. I like to teach redundantly. I think that teaching redundantly is a good idea, because if I don't get to you one way, I'll get you another. All right, so you'll actually understand this. All right, we're going through some tough times now. All right, especially with teaching. All right, the work that I'm putting in to teach now has got to be at least 5 times what I would normally do in a regular classroom, because I have to produce these videos online, and all of your exams are online, and it's not easy. Believe me, I work all day just doing this. All right. So DNA ligase, enzyme that joins the 2 DNA molecules or nucleotides by covalent bonds. This will be the 3rd time I mentioned that. And then we have something that I was going to put on your exam, but I'd rather teach this up close and personal, where I have a chalkboard or a whiteboard. But reverse transcriptase, all right? That's a pretty interesting enzyme, all right? And you know it's an enzyme again because it ends in A's, ASE. And these are enzymes that make DNA strands from messenger RNA strands. And by now you should know what a messenger RNA is, all right? So it's actually, you can go backwards, where you find a, and this is so important in medicine, all right, where you could take a messenger RNA, which you know is going to be translated into a protein, all right? And if you found a protein, let's say, for example, that messenger RNA was being expressed in a cancer cell, all right, you can go backwards with that and find out what part of the DNA, in DNA language or in DNA code, that cancer resides in your DNA. So, but I'm not going to talk much about this, because I'm not going to put this on your exam. It would be better if I was in front of you in a classroom where I can answer questions to the whole class about reverse transcriptase. Let's see, we got 26 minutes, 27 minutes on the clock. I want to go over one more thing before we go any further, and this is something that will be on your exam 3. All right, and this has to do with gel electrophoresis. All right, and gel electrophoresis basically is almost old-fashioned now, but it's still being used. All right, I did a lot of research for the Department of Justice. As a matter of fact, they funded my research when I was writing my thesis. And gel electrophoresis sorts DNA molecules on the basis of their size and charge. All right, so I may as well tell you right now, the charge of a DNA molecule is negative. So it should stand to reason that if you have a negative charge on a particle, it will be attracted to a positive pole, all right? So what we do is we have this agarose gel, right? An agarose gel is basically derived from seaweed, and we have these wells, all right, which are molded into the agarose gel. Over here, not shown, and it's not, is what's called a buffer solution. That's what you're seeing here, right? A buffer solution. And what that is sort of like the solution that's in your battery in your car, right? If there's no what they call water in your battery won't work. And this is water, but it's more than that. There are electrolytes in the water in the battery, and there's electrolytes that are in this buffer solution, right? So we have this gel, and this gel, if I was in class now, I would draw a picture on the board and show you that it's derived from seaweed, but it has these little kind of like convoluted tubules in it. They're all convoluted, they're all twisted, all right? And once we take a genome and we digest it, and now a new term comes up, digestion. Digestion in this term does not mean eating or anything like that. It simply means to break things up. And that's exactly what we do when we do digest food. We take macromolecules, which we're going to be talking about soon enough, and like, for example, a hamburger, a piece of meat, right? We have to get those into very small molecules to get them into our blood. But we'll talk about that later when we do the digestive system, all right? So what we could do is we could take a piece of DNA and we could break it down with restriction enzymes into small fragments, all right? And then what we could do is we could turn on the power supply, and we would put the positive end here, because we want these fragments to travel from here to here. Now, the larger fragments, that is to say the larger pieces, and if you read the textbook, you'll understand why this is important. The larger pieces take longer because they have to travel through all of these little convoluted tubules before we shut the power off. So the larger pieces are going to remain further over here, and the smaller fragments, like 4, 5, 8, 10, 25 base pairs, will travel the furthest, right? Then when you shut off electrophoresis, you can actually see what are called bands in the agarose gel. So what I do want you to know, especially for the exam, that gel electrophoresis sorts out DNA molecules on the basis of their size and charge, all right? On the size and charge, which I just explained. During the process of electrophoresis, the gel, which again is made of agarose, all right, which is derived from seaweed, all right, functions like a thick filter or a sieve, separating the samples according to their size. Again, the larger pieces will remain back here, and the smaller sizes will go forward until you shut the electrophoresis apparatus off. I think we'll stop here, and I'll publish this, and you guys should look at this. You should look at the textbook. You should look at the slides. You should listen to the lecture. You should take the practice quizzes, all right? Now, there's no penalty if you get the practice quizzes incorrect, all right? I am not grading your practice quizzes, all right? The reason is because I'm trying to keep you guys up with your grades as long as you do the work, all right? I'm going to pretty much base your grades on your lecture exams, plus the information I had before we had to leave the classroom, all right? And you're doing some lab work online, which I am not supplying. Another professor is doing that for me, all right? And I'll take that into consideration. But please make sure you do all the work that's necessary. Again, you all have my home phone number, all right, which I'm not going to say now because this is going to be published on YouTube, all right? And you are welcome to call me up until 10, 11 o'clock at night, all right, if you have a question. I prefer you do not text me because, you know, I got to sit there and do this. You know, I have like 50, 60 students calling me up or texting me, and I got to sit there and do this all day when I could just simply answer your question very simply on the phone. All right, so with that, study hard, and we'll get through this together. Adios. Where is this thing?